In the perpetual dance between humans and viruses, a new player has emerged from the shadows, a microscopic entity with the potential to reshape our world and challenge our understanding of infectious diseases. Discovered in the depths of Chinese caves, a new bat coronavirus, designated HKU5CoV-2, has sent ripples of concern through the global scientific community. This discovery, a stark reminder of nature's boundless capacity for adaptation, has rekindled anxieties about the ever-present threat of animal-borne diseases. HKU5CoV-2, a close relative of the notorious SARS-CoV-2 virus, has ignited a firestorm of questions about its potential to infect humans, its virulence, and the specter of another global pandemic. The scientific community is on high alert, mobilizing resources and expertise to study this new threat. The world watches with bated breath as scientists race against time to unravel the mysteries of this novel virus. Will it be a fleeting threat, confined to the depths of its bat reservoirs, or will it mark the beginning of a new chapter in the ongoing saga of viral pandemics? The uncertainty looms large. Only time and rigorous scientific inquiry will tell. The stakes are incredibly high, and the world is holding its breath. The emergence of HKU5CoV-2 underscores the interconnectedness of all living things, a sobering reminder that the health of our planet is inextricably linked to our own. Human activities have far-reaching consequences. The discovery, a testament to the tireless efforts of scientists working on the front lines of disease surveillance, serves as a clarion call for increased investment in research, public health infrastructure, and global cooperation. The need for a united front has never been more apparent. The stakes have never been higher, and the world must rise to the challenge. Coronaviruses, a vast and diverse family of viruses, are responsible for a wide range of illnesses in humans and animals, from the common cold to severe respiratory syndromes. These viruses have been around for centuries, silently evolving and adapting to their hosts. These microscopic invaders, named for the crown-like spikes that adorn their surfaces, are masters of disguise, constantly evolving and adapting to new hosts. Their ability to change and adapt makes them particularly challenging to control and predict. Their genetic material, composed of RNA, is highly prone to mutations, allowing them to readily jump species barriers and exploit new ecological niches. This high mutation rate is a double-edged sword, enabling rapid adaptation but also making it difficult to develop long-lasting vaccines. This remarkable adaptability makes coronaviruses a constant threat to global health. Their ability to spread quickly and adapt to new environments means that they can cause widespread illness and even pandemics. Among the most notorious members of the coronavirus family are SARS CoV, the virus responsible for the 2002 to 2003 SARS outbreak, which caused severe acute respiratory syndrome and led to significant global health concerns, and MERS-CoV, the cause of Middle East respiratory syndrome. This virus emerged in 2012 and has since caused numerous outbreaks, primarily in the Middle East. These viruses, both originating in bats, wreaked havoc on human populations, highlighting the devastating consequences of zoonotic diseases, illnesses that can spread between animals and humans. The close interaction between humans and wildlife increases the risk of such transmissions. The emergence of HKU5CoV-2, another bat-borne coronavirus, has reignited concerns about the potential for these viruses to trigger future pandemics. The ongoing surveillance of wildlife and early detection systems are crucial in preventing such outbreaks. The study of coronaviruses is a race against time, a constant pursuit to understand their evolution, transmission patterns, and pathogenic potential. Researchers are continuously monitoring these viruses to predict and prevent future outbreaks. Scientists around the world are working tirelessly to develop vaccines, antiviral drugs, and diagnostic tools to combat these elusive foes. Their efforts are crucial in mitigating the impact of current and future coronavirus outbreaks. The battle against coronaviruses is a global endeavor, requiring international collaboration, data sharing, and a commitment to public health preparedness. Only through united efforts can we hope to stay ahead of these ever-evolving threats. The discovery of HKU5CoV-2 in Chinese horseshoe bats has sent shockwaves through the scientific community, highlighting the intricate and often perilous connections between wildlife and human health, raising alarms about its potential to infect humans and the broader implications for global health security. This novel virus, a close relative of SARS-CoV-2, has demonstrated an alarming ability to bind to human cells, a crucial step in the process of viral infection. The binding process is a key determinant of a virus's ability to jump species. While there is no evidence of human-to-human transmission at this time, the virus's ability to bridge the species barrier has raised concerns about its pandemic potential. This potential necessitates a proactive approach to monitoring and research. HKU-5-CoV-2, like its notorious cousin SARS-CoV-2, utilizes the ACE2 receptor on human cells as a gateway for infection. This receptor is a critical entry point for the virus. This receptor, found on the surface of cells in the lungs, heart, kidneys, and intestines, acts as a docking site for the virus, allowing it to attach and gain entry into the cell. The widespread presence of ACE2 receptors in vital organs underscores the potential severity of infection. The virus's ability to bind to ACE2 with high affinity suggests a potential for human infection, raising the specter of another zoonotic disease outbreak. This high-affinity binding is a red flag for virologists and epidemiologists. The discovery of HKU-5-CoV-2's affinity for human cells underscores the urgent need for continued surveillance, research, and public health preparedness. Vigilance and readiness are key to preventing another global health crisis. Understanding the virus's transmission dynamics, pathogenic potential, and susceptibility to antiviral drugs is crucial for mitigating the risk of future outbreaks. This knowledge will inform the development of effective countermeasures. The world is watching, and the scientific community is working tirelessly to stay one step ahead of this emerging threat. Collaboration and innovation are our best defenses against the unknown. All right, let's dive into how HKU-5-CoV-2 actually infects human cells. This virus, like many others, uses its spike proteins to latch onto receptors on the surface of human cells. Once attached, it can enter the cell and begin to replicate, leading to infection. It's a complex process, but understanding it is crucial for developing treatments and vaccines. Now, let's compare HKU-5-CoV-2 with SARS-CoV-2, the virus responsible for the COVID-19 pandemic. Both viruses belong to the coronavirus family and share some similarities in structure and infection mechanisms. However, there are key differences in their genetic makeup and how they interact with human cells, which can influence their transmissibility and the severity of the diseases they cause. Reflecting on the legacy of SARS-CoV-2, it's clear that the COVID-19 pandemic has taught us many lessons. One of the most important is the need for constant vigilance. We must continue to monitor emerging viruses, invest in research, and be prepared for future outbreaks. The impact of SARS-CoV-2 on global health systems has underscored the importance of readiness and rapid response. Finally, let's talk about research, surveillance, and preparedness. Ongoing research is essential to understand new viruses like HKU-5-CoV-2. Surveillance helps us detect outbreaks early, and preparedness ensures that we can respond effectively. By investing in these areas, we can mitigate the impact of future pandemics and protect public health. It's a continuous effort, but one that is vital for our collective safety.